Hello, everyone. Welcome back. Well, welcome back because I forgot to hit record on the Zoom session. So I have to do this again by myself. Okay. So today we are talking about first few things. Before we begin, a few things. Pre-lab quiz 9. Please take that um, before you do the lab because you cannot access a video about it. Um, the lab report for experiment 1 is due today by 11:59 p.m. Make sure to submit it on Gradescope um, because that is what you're going to be graded on, and anything submitted afterwards is considered late. So there has been some confusion, I think, about the what is actually due for the lab. What are we being graded on? Okay, so you watch the lab video, but the things that are part of your lab grade are the pre-lab quiz and the lab report, because you can watch the lab video and just submit it at the end, but that's part of the lab experience. So you should watch the videos, because if you don't watch the videos, I don't think there's a way that you can complete the lab effectively. Um, and remember, you must be turning your own work. So watching the videos is key. Attending these discussion sections is important too, because I explain the theory, the concepts, and break it down for everyone um, to understand and to kind of connect the concepts together. So please, what's being graded is the lab report, because that's what requires your critical thinking skills um, to answer some questions after having gone through this experiment. Okay, so those are the things I wanted to get out of the way. If there's any other questions, always send me email or post something on Piazza. I will most likely look at it. Okay, so let's move on here. Let's move on to the experiment. So we're going to be talking about the separation of two dyes using a technique called column chromatography. Column chromatography is a physical technique, so it's something we do to we're trying to separate two compounds. We're not trying to do a reaction or anything. So column chromatography is not a reaction, because we're not changing the identity of our compounds once we put it through the column. Okay, so a little history lesson. Dyes has been around for a long time. Historically speaking, dyes before we made dyes were actually extracted from plants found by nature, so natural dyes. So first dyes that we encountered in the early 1400s or early like 15th century, we they were natural dyes, and then soon, not long after this time, we moved on to synthesizing more dyes, and that's when organic synthesis started to kind of boom and become a very important profession and topic, making new chemical compounds that would soon, be found everywhere, accessible to everyone. Making these compounds is what pushed organic synthesis to the forefront of the chemical sciences, um, as as one integral part of chemistry. Okay, so that's all I wanted to say about this image. But this is a picture I found. Dine Wolkoff, and I think it yeah, this guy Bartholomew Anglicus, he was in Paris, and so this is when I think they actually investigated um, or talked about first dyes when they first encountered them. Okay, so dyes usually are made. We actually make them from petrochemicals or uh, petrochemicals, or what we call like chemicals obtained from like fossil fuels. So many of the products we see today in everyday life, they come from petrochemicals, including dyes right here. So dyes is another important commodity, a material that we use, or well, not material. So my bad, not that material, but it's a product we use to you know to make clothing, to dye clothing, make all these colorful clothing items. So without dyes, we wouldn't have very much option for clothing and other stuff. So our personal fashion sense would be kind of boring, because there'll be a limit of colors to kind of wear and use for clothing. So dyes gives us a variety of colors, so we can make and create more beautiful items, clothing, um, and pottery. So dyes are important for color. So without them, we would have not really much color options. Okay, so here is a historical collection of over 10,000 dyes. So there's lots of dyes, and they make up a whole spectrum of colors. So you can use a particular dye for one color, and then or another dye for another color. So we can get a whole library of different colors, and that gives us much more options when we're coloring things, and when you know making objects different colors. So um, the number of dyes you can actually make or create is endless, because in organic chemistry synthesis, you know what they say, the sky's the limit. Unless you try, we can make it, right? We could probably make it. But that's why organic chemistry is always an evolving field of study and discovery. Okay, so historically, dyes came from plants, but there are disadvantages with doing that, harvesting them from plants, because they require a lot of land, water, uh, and other intensive use of resources, which is not very sustainable, um, or it's not sustainable, and it's not green, and it's energy intensive, and it could be energy intensive. It probably is, if you have to like cultivate plants, harvest them, and extract the dye compounds out of the raw materials. So we also don't want to use fossil fuels, because they are not very nice to the environment. So fossil fuels are not carbon neutral, meaning in the carbon cycle, once we burn fossil fuels, the carbon dioxide that's released stays in the atmosphere. It's always increasing, because once you use the fossil fuel, it's gone forever. You can't unburn something, right? The fossil fuel, you can't unburn it. So the carbon dioxide will always go up into the atmosphere. But with that's why we go to renewable resources like biomass, because living organisms like plants, they trap carbon dioxide, and then we can in turn extract that that carbon or that organic matter from the plant and use that material we extract to convert it to other compounds. And then once we plant more trees, um, plants, or any kind of like plant any kind of organism with plant cells, or like shrubs, trees, grass, um, wood. Basically, if we replace trees, we can absorb the carbon dioxide again from the atmosphere, and then recycle that plant when at the end of its lifetime. Or if we dedicate trees to for as a renewable energy source, we can always go through that cycle of recycling the carbon, so it's carbon neutral. So in the end of the day, we can use that material in a cycle, and that's how we kind of can reduce the effects of climate change, because we're not continuously pumping CO2 in the air by burning fossil fuels. But with living organisms like trees, they can be replaced because you can just plant new trees. Um, so that's a way of using biomass to kind of replace diamol to as a renewable resource to make compounds like dyes or other useful uh, other useful compounds for other parts of industry or society as a whole. So I bring this up because in my research as a grad student here, I actually worked on converting compounds obtained from cellulose, which we got from trees, and converting them uh, into dye molecules. So we actually made this dye molecule not from petrochemicals. We didn't extract it from plants. We actually took just raw cellulose and converted. We took a compound called CMF, which is 5-choral methyl furfural, and we took that compound right here, that's CMF, along with angelicolactone right here, that's a lactone, and we condensed them to form this dye molecule here, which has an extended pi system. See the pi system here, conjugated, double bond, single bond, double bond, single bond, double, single, double, single. So the conjugation extends across the whole molecule, creating a chromophore, or what we say, a part of the molecule that absorbs light and emits light in the visible region. So this is the color. This compound, when we dyed fabric here, this thing in the background is fabric. So when we dyed it, it produced this yellow color. So this compound is effective as a dye. So nowadays for green chemistry, we try to make useful chemicals that were previously like only accessible from petrochemicals, like industrially possible. So now we're moving towards the use of renewable resources such as cellulose, because they are renewable and sustainable. And once you use them, you can recycle it back, because we can always use more we can always when trees age, they produce mass or biomass that just gets left over, right? So but if we can convert it, we can actually convert all that dead matter and convert it to something more. And then we can in order to keep the cycle going, we could just replace the tree, and that will create a cycle that is net carbon neutral and therefore not contributing to global warming, which is what happens when you have CO2. CO2 being continuously produced by burning fossil fuels. Okay, so enough of rambling. So this is my work that I did, I was a part of. And here is the paper, in case, or the link to the paper or information of the paper if you're interested about reading sustainable synthetic dye compounds from compounds that we obtain from nature. Okay, so usually if a compound is part of a part of a natural reaction or natural route, that is usually more sustainable than uh, burning fossil fuels. Okay, so characteristics of dye compounds. So you guys are learning, or everyone's learning probably about aromaticity and conjugation in lecture. So this kind of ties in with that. A dye compound has conjugation across all the all conjugation across the whole molecule. So what that means is that there's a alternation of single and double bonds within the molecule. So right here, it could be in a ring, but in this case, it's conjugated all over 3 rings through the central carbon atom right here. See double, single, double, single, double, single, double, then single right here again. And then we can see that going through the bottom in this ring for malachite green. Crystal violet has a similar structure. And then this is also possible in linear molecules. See where the conjugation expands across the whole molecule. Throughout the whole molecule, we have this alternation of single, double, and single bonds all across the molecule. So what that does is it allows the energy levels for the electrons, the electronic energy levels, to be lowered. So when you excite an electron, it'll absorb and emit light in the visible region because it doesn't take much energy to excite that electron because the energy levels are much lower than a compound that has no conjugation or has little conjugation. So dye molecules or things that are colored usually have energy levels that are really low with each other. And then other compounds like benzene or like compounds with no conjugation like ethene have energy levels that are pretty much much larger electronic energy levels. So the energy that's required to excite them, it's going to be much more than visible light. So it'll require radiation or energy or electromagnetic radiation or light from. It'll require a much more higher energy form of light. So electromagnetic radiation, it'll probably be in the towards the UV part of the electromagnetic spectrum. So stuff we can't see and that are very high in energy. So with the conjugation, you tend to get colored compounds. So we have like orange fruits, orange vegetables, carrots, mangoes, pumpkins, papayas, cantaloupes. So that is where beta carotene. So this long molecule is called beta carotene, and that's responsible for the orange color in these fruits and vegetables. Okay, so that's all I want to talk about. So that's how dye compounds work. That's why they're colored, and that's why this things you see that don't have this extended conjugation, that's why they're not colored. Okay, so in this lab, you'll be exploring crystal violet. Here's a picture of it in aqueous solution, and here's a picture of it as a solid. And see how they're different colors in the solution as a solution and as just the raw material. Malachite green, very nice picture taken by Loco right here. Very nice. So malachite green has a very nice forest green color, and that's due to the conjugation we see here. Okay, so I'm going to talk about chromatography. So bear with me. There's a lot. But it helps explain what chromatography is in a nutshell. So chromatography is the technique of separating 2 compounds in a mixture, and usually this mixture are both. It doesn't have to be liquid, but it can be liquid. Usually, it's for a mixture, like a mixture of 2 liquids. Sometimes you have to separate a mixture of a solid. You can use chromatography for that, but sometimes you try to like separate the two components using filtration, recrystallization. That's usually another good way to recrystallize to purify solids. But usually chromatography, you can, if you can't purify by recrystallization, you would use chromatography because it works for every kind of mixture. But we never work with a mixture of gases, so that's something we never usually work with. Okay, so this is a very important skill in organic chemistry, or in basically in chemistry in general, because when we do reactions, we need to purify it before moving to the next step. That's usually the case. So we usually purify it by using column chromatography. So the dyes, they're going to be easy to separate because they are colored, and they are colored, and we can actually see them separate on the column. So that's why dyes are very fun to work with. So chromatography refers to the separation of a mixture of compounds based on their polarity or their affinity to stick to a stationary phase or solid. And the stationary phase can be polar or non-polar. In this case, it'll be polar. You're going to use alumina in the stationary phase. There's the other type of material we commonly use for column chromatography is called silica gel. But in this case, we're going to use alumina. Okay, so the two compounds, they'll have different polarities and they'll interact with the column at differently. So that's how we will be able to separate them, because one will interact it with more strongly and one will have a lesser affinity, so it'll be able to move down the column more easily. So how can we pass a mixture or liquid through a solid by gravity? Well, the thing is, silica gel is actually, sorry, not silica gel. The stationary phase alumina will have, it's porous, so it has spaces in the material for small molecules to pass through. So we let the compounds flow through the column, and eventually at the end, one compound will come out first. You collect it, then you collect the second compound, and at the end, you have separated the two. So in this case, we're going to use two different kinds of solvents to help separate each component in sequential order. Okay, so let's talk about what this means using the diagram. Okay, so first, let's just take a typical look at what a chromatography is. So you see here that chromatography has been around for a long time. It's the thing about chromatography, though, it takes a long time to, it takes a really long time to do. It can be time consuming, and it can be tedious. So nowadays there's automated columns, so it makes this process a whole lot faster. But when I did columns, I prefer to do columns by hand, so the old school way, and they can easily take up to 2 hours. So you would run a reaction, purify it by chromatography, and then call it a day. So there's that routine of like in chemistry, you're just doing a procedure over and over again until you get what you want, and keep on changing parameters, always looking at experiments from different angles to see what went wrong. So chemistry is always it's a process. It's always a process. You will never get results quickly. I don't think you'll get them quickly, because I think to be able to do chemistry, you need a little bit of luck as well. But that's just a background, like what organic chemistry is. Organic synthesis, or OChem in general. Synthesis and chromatography is something that comes hand in hand. You can't avoid it. But nowadays, if your lab can't afford one, usually they'll try to do it automated column chromatography. But usually that comes with a price. So usually most labs probably can't afford it unless a department has a dedicated automated system. But I wouldn't trust the system, because I'm afraid I might break it, so I always prefer to kind of do my columns by hand. It's a good, it's a very good technique to know. It's something I have not myself mastered because I rarely did any columns um, during my time here. But anyway, so these are what columns are. See the large column. Of course, the column you would use is not that big. This is for like industrial scale. Well, not really industrial scale, but just because this looks like a lab. So this is actually probably this is probably when you're trying to isolate it, I would say like maybe 100 grams or close to less than 100 grams. See, here is our stationary phase in the column here, and he's pouring his material on the top to load it, or right, or that could actually be solvent. I think that's solvent for the column. And when we go through the column here, at the bottom is where you'll collect the liquid that comes out. So usually your compound will come out in the solvent. Um, and then to collect your material, you would evaporate the solvent at the end. Okay, so here is column chromatography. It's a very time-consuming process. So you would load your stationary phase, then load your solvent system. So the first solvent you're going to use to push your materials through the column. Then here is your material that you'll load at the very top of the column, right here, where the liquid level is at the very top. So it'll load safely. You want to load it on the top of the column because if you load it in a bunch of liquid, it'll be dispersed, and you don't want that because it'll be hard to get off the column as you go through this process. So column chromatography is an art. It's an art. It's like sculpting because you care about every process. So you have to do it correctly every step of the way. So you will load your material on the top of the column right here. So loads at the very top where the solvent level is. So it can easily go through onto the column. See here, as we add more solvent here and start the column, you'll see that if it's colored, it'll separate into bands, and that tells you that one compound is traveling faster than the other, and one compound is adhering to or absorbing to the column much more strongly. And that's how we are able to eventually separate until this goes all the way to the bottom and comes out, and you collect it. Then you collect the blue compound, the other compound in your mixture, and then you're done. And then that's the end of column chromatography. So then you would clean up. So for dyes, this is very, I think it's very relatively simple. It takes a lot of work, but it's simple to do. Now, if you don't have two colored compounds, that's a problem because you can't see them, so you don't know when each of them comes off the column. So that's a lot harder, and it requires another analytical technique to determine where your compound is. So typically, if you have colorless compounds, it's a lot more work and a lot more time consuming. I would say that's why I say that's why when I say columns are time consuming is because it can take up to hours depending on how much material you have. So it's better to work with less than more because it'll take less time to do. So here is an example here's an example I think by a student who kindly took photographs of the whole process. As you see here, as we load the material onto the column, it will slowly distribute into bands, and those bands are probably different compounds in the mixture. So we can use and the colors to see when each compound will elute out of the column. So this is how column chromatography will work. See how the bands they are traveling at different rates down the column. That means they adhere differently to the stationary phase. So we always choose different solvents because we want to get the material off the column eventually. So we choose a solvent that will kind of have a better affinity for one compound and that will elute first, then use a different solvent to get the other compounds out. So it's always the interplay between solvent polarity. So you're always choosing different solvent ratios or solvent systems to affect the polarity of the solvent, and that will help you elute compounds off the column. So in this case, I use one solvent, it'll elute my yellow and orange pretty well. And then I change a different solvent to elute the red and red orange components more faster. So this is a typical example of column chromatography. So usually in a mixture like this, you're only looking for one compound. So you would actually want to test this first to see if it's your compound or if you know it's your compound. And then, of course, there's tricks to help you along the way with column chromatography, but that's not the point of this experiment. In this experiment, we're going to use a solvent first to isolate malachite green, then use a different solvent to push out the remaining crystal violet. Okay, so that is it, everyone. Hopefully the experiment goes well. So watch the lab video. Because if you don't do the lab video, I do not know how you'll answer the post-lab questions. So watch the lab video. Watch the lab video, answer the questions so you understand how the lab works. So then you can answer effectively the post-lab questions. It'll take less time. You'll spend less time on the post-lab questions so you can focus on your other classes and stuff in the lecture. So that is it. I'll see you all next week. Have a great weekend and stay safe. All right. Bye, guys.